So good afternoon and welcome everybody. We are very fortunate today to have Professor Usha Vijayraghavan with us. Professor Usha Vijayraghavan did her PhD from Caltech with Professor J. Abelson, and her postdoctoral research was at Caltech again with Professor Elliot Meyerowitz, who, as you know, is very famous. There she began her work on transcription factors that regulate plant development. She became a faculty at the Indian Institute of Science, Bangalore at the age of 29. You heard me right, at the age of 29. And she has been there ever since. She holds one of the 3 endowed professorships at IISc, the J. N. Tata Chair. And she is also Dean of Biological Sciences at IISc. Incidentally, she is the first woman dean at IISc. Her group uses genetic and genomic tools to investigate key factors that control rice plant development. Her group has also made important discoveries on transcription factor-driven complex gene networks that control plant panicle architecture and floret fertility. She has many fellowships and awards to her credit, like the J.C. Bose Fellowship, as well as fellowships like the Rockefeller Foundation Fellowship, the Wellcome Trust International Fellowship of UK, the NBIOS Awards, the C.V. Raman Award, DBT Bioscience Award, etc., etc., etc. She is a fellow of the Indian National Science Academy and the Indian Academy of Science, Bangalore. Her scientific expertise is sought after as she serves in various advisory committees in India and also internationally in leading institutions and granting agencies. So welcome, Professor Vijayraghavan. We look forward to hear more.
Good afternoon, ladies and gentlemen. It's my distinct pleasure to be present here at Bose Institute. And at the outset, I should thank Director Bose Institute, Professor Kaustubh Sanyal, the Bose Institute Colloquium team in thinking about my work and my team's work and giving me this opportunity to give you a sort of bird's eye view of the big questions that we are trying to address and trying to understand. I am absolutely consider this a big privilege to be talking at the Bose Institute and delivering this seminar. So given that the audience might be diverse and I believe this has also been put in the Bose Institute YouTube channel, I'm keeping the first part of the talk general to give you a very, very broad description of the problems which are exciting to me and hopefully exciting to the rest of you also. And then I will also go technically a little deeper into on 2 different topics which we are focusing on recently and leave some time for questions. Okay, so when we look at life on Earth today, there are many, many forms of life on Earth, but perhaps the oldest and the most earliest detection of multicellular life forms arose about 2.7 billion years ago. I think I can't use a pointer here. Right. Yeah. So I'll either use this or this. So about 2.7 billion years ago, and that we can trace to the origins of the first eukaryotic cell. So the eukaryotic cell, for those of you who are from physics, chemistry, math backgrounds, are the ones which have compartmentalized their functions to represent the hereditary material in the nucleus, the energy metabolism, etc., in the mitochondria that happen through the acquisition of another single cell prokaryote. And for quite some time, this lineage, which is marked in brown, was the common ancestor for both the plant and the animal kingdom. It is about 1.6 billion years ago that the two major kingdoms of the multicellular eukaryotes split into the plant lineage and the animal lineage, and this occurred again through the acquisition of another prokaryotic symbiont, which later on became an organelle, and that gave you the plant lineage. This is because of the acquisition of a new organelle called the chloroplast. Right? So the origins of plants and animals as multicellular models for studying developmental biology actually trace back to very, very ancient times. That is about 1.6 billion years ago. So the general broad questions that one can ask in developmental biology is, how do you go from a single cell zygote to a multicellular organism with tissues and organ systems which have distinct functions? So that's a broad question that transcends both the plant and the animal lineages. But as we all know, plants are not animal-like, whether it is at the cellular architecture or the tissue architecture or their physiological, etc. So therefore, while the broad rules of becoming multicellular and acquiring different traits might be similar, the actual toolkit or the molecules which are used to undergo development and differentiation are likely to be different between plants and animals. So even amongst the plant lineage, in today's talk, I will restrict myself to talking about one group of plants which are called flowering plants, those which make flowers in which the reproduction occurs. And these are specialist organs seen only in this group of plants which are called flowering plants or angiosperms. Interestingly, flowering plants or angiosperms are the equivalent of mammals of the mammalian clade. They are rather recent. If you look at the history of the plant kingdom on planet Earth, they have been on Earth for only about 200 million years, as opposed to the billion years since the plant lineage had been originated. So if you look at how flowering plants are made, they are also made from an embryo, and that embryo, it comes from the fertilization of a single egg cell by the sperm nucleus, which is the male gamete. So you have a male gamete, you have a female gamete, just like in the mammalian development, these two fuse and give you a single cell zygote. That then makes an embryo. And initially, the embryo is just a ball of cells, which is attached to the maternal tissue by a linear set of cells, which are called the suspenser. So unlike the mammalian, this then develops a little bit further to give you something which has got two rudimentary organs called the cotyledonate leaves. And in between them is a small niche of cells, which is called the shoot apical meristem. It makes a somewhat ground tissue over here. And in the opposite end, there's something called the root primordia. So the angiosperm embryo has a very, very simple structure. It has axis developed in the north-south, what we call as apical basal axis, and it has the round or the horizontal axis developed by the epidermis in the internal tissue. That's all that there is in the embryo which is present in the seed. This structure then goes on to giving you all the organ systems of the plant. So much of plant organ system development is post-embryonic. After germination, structures are made from the shoot apical meristem, structures are made from the root meristem, and that gives you the adult plant structure. Contrast this with a vertebrate embryo, whether it is a mouse or whether it is a fish or so on. Embryogenesis is when organ structures are made. And post-embryogenic development of a vertebrate embryo, whether it is a human or a mouse, is very limited to growth in size or sculpting of tissues and structures and so on. So plants offer an excellent method by which one can understand how tissues and organ systems are made throughout the life cycle of an organism and not restricted to embryogenesis. So you have the apical basal axis and you have the radial axis, that's all there is. And it is from this group of cells, which are the shoot apical meristem, that you generate all the above ground structures of a plant. What are the above ground structures? They generate an internode of the stem, a leaf, and in the axil of the leaf will be a small group of cells, which are called the axillary meristem. which also have the potential to form a second growth axis. And this modular structure keep on repeating itself till the plant is growing in this fashion. It's called vegetative development because all the organ system that it makes are all green and they are specialized for photosynthesis. The photosynthetic organ is your leaf, which is a flattened, dorsolaterally flattened structure, um, maximized for light capture and optimized for conversion for primary fixation by using light or sunlight as the energy. The below ground system, which I have not shown over here, all arises from the root primordia, and that gives you the structures which go below the ground, form a complex root system that is required for nutrient and water acquisition from the soil. So this plant is then said to be growing vegetatively, that is producing more leaves, more stem, more buds, and so on. When the growth has achieved a certain level of growth and the time is right, then this same plant will then transition to start to make these new structures called flowers. And these flowers did not exist before. What are these? These are specialized organ systems that do not occur earlier in development. And these organ systems include the formation of the stamen and the carpel, which are the organs where the gametes will be produced. And those gametes will facilitate the next life cycle after germination takes place. So you can see throughout the development, there is a huge amount of plasticity. How to produce an organ, where to produce an organ, what should be the fate of the organ. All of these are questions which are open to experimentation for fundamental knowledge gain. And since plants are the sustenance for carbon fixation for the earth, producing biomass, in addition for giving us a lot of edible material, not only us, for the animals and livestock, they are pretty much essential for life on earth. Right? So there are many inspirational ways by which one can use flowering plants as a model. And this has not only inspired scientists, including the iconic people like Sir J.C. Bose, it continues to inspire people from all domains of life. People have admired how flowers are made, how diverse they are. Poets have written about them, painters have painted them, and philosophers have written about them, including Charles Darwin, who actually wrote a book called The Abominable Mystery of Flowering Plants. How did they suddenly come? And they appeared on Earth within 200 million years. Before that, they were all non-flowering land plants, whether it's gymnosperms or sperm, bryophytes or mosses or liverworts, that's all there was. In 200 million years, we got an explosion of the flowering plant lineage, and they occupy all ecosystems that are there on planet Earth. They all have one common property. Reproductive organs are positioned within the flower, but the flowers themselves have a diversity of organ shapes and colors and positions, but they have certain commonalities. I'll come to that in a bit. So flowering plants in this short bit of time actually split into two major lineages about 120 million years ago. They split into one large lineage, which is called monocots, and another lineage, which are called the eudicots and the basal dicots. So they differ a little bit in their anatomy and structure, but this is an important split to keep in mind because it is in the monocots which are embedded the family of grasses where all the cereals come from. So our nutrition, our caloric intake, everything comes mostly from that of the monocots. Whereas all our vegetables and fruits, crops, and flowers which we admire and look at, most of them come from the dicot lineage, right? So what are the big questions that one can ask? I just told you that plant development is very plastic. That means it germinates, a seedling germinates, gives you vegetative organs, and then depending upon the conditions, which are both environmental as well as genetic, encoded within its own genome, it will then switch to start making reproductive organs. So one can start asking questions of what are the triggers which convert the shoot apical meristem from making leaves and branches to now start making these new organs which are called flowers. Then one can ask, what is so special about the flowers? What type of structures do they have? Why and how do they facilitate the formation of the gametes and the next generation? To ask these big questions and to understand how genes and the interaction with the environment drives development, you need a model system. And that came from the discovery of this weed, the weed that inspires you in the lab and not in other places. This weed is literally a weed. You would find it anywhere in the temperate zone, and it's called Arabidopsis thaliana. And Arabidopsis thaliana is a close cousin of mustard. It's of no direct value to humankind at all, other than the fact that it has exploded our understanding of plant developmental biology. Now, why is this Arabidopsis thaliana such a big deal? It is a big deal because its genome is the smallest. That is, its inherited DNA in the nucleus, which is capable of having the entire blueprint of germination, vegetative growth, reproduction, flowering, seed formation, everything is packed in the smallest amount of content which is present in the nuclear genome. And that content is just 5 times that of the single cell organism budding yeast, Saccharomyces cerevisiae, which grows, or it's also, or the brewer's yeast and so on. So in the smallest genome, it encompasses all the properties of a flowering plant. And it has a very short lifespan. It can go from seed to growth to producing the first seed in about 6 to 8 weeks. So it's a short lifespan, so one can do genetics. And the combination of having a small genome and short lifespan, the ability to make mutants and understand how mutants disentangle a developmental process made Arabidopsis to become the model for understanding plant developmental biology, particularly for flowering plants, just as Drosophila melanogaster has done wonders for us to understand how development occurs, or principles of development in animal systems. Right? And a whole lot of molecular tools, etc., came in. Very interestingly, Arabidopsis thaliana was the perhaps the second eukaryote ever to be fully sequenced end to end. After Saccharomyces cerevisiae, it was not mouse, it was not Caenorhabditis elegans, it was not Drosophila melanogaster, it was Arabidopsis thaliana, whose genome was fully sequenced. So the availability of all the tools of Mendelian genetics, modern tools of molecular biology and reverse genetics allowed people to put all the tools together to understand questions in development. So now let's come back to the question which is pertinent to today's talk, and that is on how does this small group of cells, the shoot apical meristem, that is made in the embryo, give rise to more cells which will go on to make the organ systems of the plant. So early in development, the cells in the shoot apex will be dividing, and they will give more cells that get pushed to the flanks, and from the flanks arise these new organ systems which you can see are developing as leaves. But after reproductive cues are attained, that is the right temperature, the right photoperiod, the right nutritional conditions, the right hormones are present, then this very same shoot apex now start producing from its flanks small group of cells which are not growing in a flattened manner. They are growing like a new ball of cells. So this ball of cells looks pretty much similar to the apical meristem which is growing up over here. And this region is called as the multipotent stem cells. That is, they have no identity. They divide and give rise to more cells. The cells that they are producing get pushed to the side. Plant cells are boxed by a cell wall. So when division occurs, so if I divide, I will make one more cell. So me and my mother will be right next to each other. Then the daughter will divide and the daughter will give one more. So there'll be 3 of us, and you get pushed to the side. So that's what happens. And when you're pushed to the side, it is from the peripheral regions that organ systems and organ formation will begin. So these cells are now growing to give a ball of cells on which you start seeing new organ systems whose pattern in which they emerge has changed. Floral meristems, these are called floral meristems, meaning they're going to become flowers. They arise in a spiral phylotactic on the flank of the shoot meristem. But the organ systems of the flower arrive in a concentric pattern. So there is a change in the pattern in which organ systems are arising from the meristem. These circular patterns are called whorls, and the first whorl of organs which are formed are the sepal, the second whorl are the petals, the third whorl are the stamens, and the central mound will give you the carpel. So in an Arabidopsis plant, every single flower which is made in the plant, whether it is the first flower or whether it is the last flower, whether it is the Arabidopsis plant grown in Kolkata, in Bangalore, or in Berlin, or in California, it will have the exact same pattern. 4 sepals, 4 petals, 6 stamens, and 1 carpel. Fixed and constant. So one can now start to ask, how do you define a floral meristem? How do you define the organ primordia in such a way that you get a stereotypic, tactic, invariant, species-specific formation of the floral flower structure? Right? So these are questions that one can ask using the tools of genetics. The has been approached, so you know nothing. So you want to start to ask, what are the genes that drive the process of making this invariant pattern? And this invariant pattern is also not invariant only for the Arabidopsis plant. This pattern of organs, that is sepal, petal, stamen, and carpel, is invariant for all angiosperms. That means this is a pattern that was evolutionarily designed 120 million years ago. So this is a design pattern that is so successful, it is there in all flowering plants. You always have sepals, petals, stamens, and carpels in this order only. The number may vary, the shape may vary, the position may slightly vary here and there, but the order is always the same. So using this as a model, geneticists, knowing nothing, said, let's make mutants. Let's make mutants which change the pattern of the organ. Organs are not positioned where they should be, they come in the wrong pattern. And this was taken up by Elliot Marowitz at Caltech, my postdoctoral advisor. Was also taken up using antirrhinum as a model by Enrico Cohen at the John Innes Institute. Heinz Sadler took Petunia as a model at the Max Planck Institute, Köln. And at the Wageningen, Martin Corney used Arabidopsis as a model. So using 3 different families, but same approach. Can we make mutants which disrupt the process? And then from the mutant, we will work backwards and infer what is the gene in which the mutation caused the phenotype. So shown here is an example of a mutant in just one gene, agamus. And in the agamus mutant, what you're missing is the ability to make the stamens and the carpel. So you have your normal round of sepals, you have your normal round of petals, but instead of making stamens, you make another round of petals. And instead of making carpel, you make another round of sepals. So a single gene mutation has completely disrupted the process of making stamens and carpels. You cannot make them anymore, so no more gametes, and the flower is called agamus. And this is an example of a mutant which is actually prettier than the wild type. Why is it prettier? It has more number of petals. So breeders and botanists have been making these type of flowers, and these are called double flowers, without knowing what the gene is. But it's only the geneticist who will start asking, what is the gene that's causing this phenotype, right? So these were not the only mutants which were isolated by Elliot and others in the world. They isolated yet other mutants which show other sets of altered patterns. And from this, they inferred a universal model, which they called initially the ABC model, and now it is called the ABCDE model and its variants. And I will simplify these fundamental discoveries made about 30 years ago. It just opened the floodgates for us to start to understand floral development in not only Arabidopsis, in other species also. So they, by looking at the mutants, conjectured that the mutants can be classified into three groups: A function genes, B function genes, and C function genes. And it is the combinatorial action of these genes that can specify the four organ types. The model from the mutant said that wherever only A function gene is expressed, you will get sepals. Where A function gene and B function gene work together, you will get petals. Where B function and C function genes work together, you'll get a stamen. Where C function gene alone is functional, you will get a carpel. The model also predicted that A function genes and C function genes are antagonistic. That means whenever you lose A function, C will spread all over, and therefore you should get a overproliferation of stamens and petals and the loss of sepals. And this and all the model was proved true. So beyond the A, B, and C, yet more other regulators, that is the D and the E, were discovered. The D function genes are required for making the ovule within the carpel that generates the female gamete. And the E function gene are coactivators or complex formation units which put the complexes together. What are these A, B, C, D, E, G functions encoding? They all encode transcription factors. So these are master regulatory proteins which act as developmental switches to turn on or turn off downstream genes that can cause development to take place. So in Arabidopsis, A function genes are represented by two genes, AP1 and AP2, B function by two genes, AP3 and PI, which heterodimerize, C function by a single gene, A gammas, the mutation in which I just showed you before. It has an overproliferation of petals. D function is represented by two genes, and E function is represented by five genes. So the combination of which class of transcription factors is present will give you protein complexes of varying nature. So this complex will give you sepals, this complex will give you petals, this stamens and this carpels. So this is like setting up a zip code within a group of cells which are normally identical. That zip code will determine what type of differentiation program should come downstream. These MADS proteins, as typical of transcription factors, have a 56 amino acid DNA binding domain, which is called the MADS domain. And in fact, it is the discovery of the Arabidopsis MADS domain transcription factors which allowed this to be identified as a very ancient DNA binding domain that is present right from the single cell yeast to the humans. So the M stands for MCM1, something that Kaustubh knows very, very well. It is involved in yeast for controlling MCM1 and MCM2. One of them is involved in mating gap switch, the other one is involved in DNA replication complex, etc. A gammas from Arabidopsis, deficiens from Petunia, and S serum response factor, which is mammalian. So this small DNA binding motif is ancient, billion years old, billions of years old. But it's the rest of the protein which gives it its functionality and so on. Right? So now that was a very, very quick primer to now what is textbook knowledge. To generate the four organ systems in the Arabidopsis flower, you need the combinatorial action of A, B, C, D, E class of transcription factors. And depending on the nature of the complex, you can either make a sepal or a petal or a stamen or a carpel. And the interactions between the A and the C genes will make sure that the stamens and the carpel come only on the inside of the flower, and the sepals and the petal come only on the outside of the flower. Now, I just told you that in flowering plants, there was a major split between the monocots and the dicots, and that itself is very ancient. That occurred about 120 million years ago. So if the ABCDE genes and the transcription factors are involved in making the dicot flowers like that of Arabidopsis or tomato or petunia, are they doing the same thing in the very, very distantly related plants which are coming from the monocot family, and specifically that which belong to the grasses or the cereals which are called as the Poaceae family or the Gramineae family? Why does one even want to think about it? We want to think about it because, number one, All flowering plants shared a common ancestor at one point. Then they diverged. So the ground truth or the ground plan, there must be some similarities. So the genes that control making the flower in Arabidopsis, orthologs of those genes must be present in the case of the grasses. Yet, when you look at the grass flower, you look at the cereal flower, whether it is the ragi flower, that is the finger millet, or whether it is rice or Rhiza sativa, or whether it is sorghum, all of those flowers are outer two organs are structurally and functionally very different than that of the sepals and the petals of the dicots. First, the two outer organs are called the lemma and the palea. They are thick, leathery organs that completely close the flower. The flower is 90% of the time a closed structure, unlike in Arabidopsis, another dicot, which is open and the petals all come out, the stamens emerge out, and it can be attractive to the animal kingdom. This is a closed structure. Second, the equivalents of the petal are two minuscule organs which are called the lodicules, and these two lodicules are present in only one half of the flower. They do not come in the concentric ring. So the structure and position of the petal-like organs is itself very different. The stamens and the carpels are present at their normal position. So I told you that the flower is mostly a closed structure, and that is because the two outer organs, the lemma and the palea, are interlocked with each other through this hooking mechanism to keep the flower closed. These outer two organs, the lemma and the palea, are also very, very unique in having this lot of epicuticular waxes and thick structures, and this goes on to form the husk of the seed. Right? So the questions that emerge are, what is the homology of these organs, these highly modified organs which are present in the position of the petals of dicot flowers? What is the homology of the lemma and the palea to the organ systems in the dicot species? Secondly, why are they so functionally and morphologically so much variant? So these are questions that arise, and therefore one can ask the question, are the Matzbox ABCDE class families orthologs present in the case of rice? Or are they a completely new gene set which is required to make these highly modified organ systems? So the answer is a mix of two. Mostly the organ systems are dictated by the presence of the ABCDE class type of transcription factors. But what has happened in the rice and the other cereals is the number of the genes for each family has slightly varied due to gene duplication and variation of function amongst the duplicated genes. For example, the class A genes in Arabidopsis are dictated by only two genes, AP1 and AP2. Whereas in rice, there are at least four of them. Right? And if you take the class B genes in Arabidopsis, it's two of them. But in rice, it is three. So there has been gene duplication that has occurred during the process of the evolution of rice. And if you look at the C function genes, it's a single gene in Arabidopsis, and it is two genes in the case of rice. Right? So why are we having these duplicated genes? Are these duplicated genes performing the same function but half the activity, so that when you combine it, you get the full activity? So both are equally competent, but they perform only half the function, or do they perform two non-overlapping half functions in order to make the carpel? So that has been addressed, and one knows that in the case of making the carpel, MADS3 is important for specifying the organ, the stamen and the carpel, whereas MADS58 is required for determinacy. That is, in the center of the flower, you should make only one carpel and no more floral organs should emerge. So there is a diversification of roles between the duplicated homologs. In today's talk, I will tell you a little bit later on these two genes, which are both related to the pistillata clade, but have diversified their functions. I also said that there are also some new genes which come into play. For example, in the case of rice, carpel development, there's one more gene which has got recruited from its role in other species where it is involved in some other biological processes that is called DL or drooping leaf. Now that gene is also contributing to carpel development. So there is gene duplication, there's diversification of function, acquisition of new genes. All of these put together are giving you the new organ system which are specific to that of the rice flower. In the first part of the talk, I will divulge a little bit more on this set of genes. Four genes together, completely redundant in Arabidopsis. That is, four genes are present, but one of them is sufficient. So they are completely redundant with each other. And what they do is they provide the platform or the complex a mediator for the other ABCDE genes. If you look at their homologs in rice, there are five of them. And very interestingly, these five genes show distinct domains of expression. They don't show the same uniform pattern of expression here. So we ask the question, if there are five genes which are showing some distinct patterns of expression, are their functions also different? So I will take the example of OSMADS1, which is this gene marked here in yellow. And when you look at its phylogenetic evolution and place it with other Sepelata genes, which are the class E genes, you see that MADS1, together with MADS5 and MADS38, is forming a distinct evolutionary branch which has deviated away from SEP4 and, of Arabidopsis. And in this branch are present only members which are from the grass family. So when you have large number of diversification, but only in the grasses, then that tells you that maybe these genes are doing something special to make the grass floral organs. And that was a big plus. And we started to ask, what is the consequence of a mutation in one of these genes, that is MADS1? In Arabidopsis, unless you mutate all four of the organs, all four of the Sepelata genes, you will not be able to disrupt floral development. So this is a quadruple mutant in which you see that instead of making floral organs which are sepal, petal, stamen, carpel, you are getting only leaf-like organs which are coming out. So this is a single flower. Instead, it's looking like a bunch of leaves which have crumbled together. But in rice, you don't need a quadruple mutant. A single mutant is able to convert all the organ systems into something which is BRAC-like. So there has obviously been acquisition of new function for this gene over its cousins, which are the MADS34 and MADS5. So this is a member of this subgroup, but loss of just this one gene is enough to disrupt floral development. But what about the loss of the other gene, its cousin? When you lose the other gene, floral organs are normal, but branching is abruptly gone. So closely related paralogous genes have gone on to acquire distinctly different functions. One of them is important for floral meristem development and for making the flower. Other one is important for inflorescence branching. So evolutionary time and gene duplication allows for closely related transcription factors to perform entirely different functions. Now, since MADS1, even though it belongs to a member of a gene family, has distinct phenotype by disruption of just a single gene, we asked where is it expressed? And we were able to show that MADS1 is expressed in the floral meristem uniformly. And as the floral organs are emerging, the lemma, palea, and this thing, its expression itself gets restricted. It's expressed in the lemma and the palea, and it's expressed in the carpel. And this carpel region is the one which contains the stem cells, and eventually this expression pattern also goes away. Now, having known that OSMADS1 is important for floral development, we then used an RNAi transgenic, which will knock down the endogenous MADS1 expression, and compared in the flowers in this developmental state. So we dissect out the panicles in which the flowers are occurring, and all of this occurs in the embedded stem. We pull out these and determine the global transcriptome in the wild type versus the mutant. So this allowed us to infer that MADS1, about 15% of the genes that it is controlling are transcription factors. So here is a transcription factor that is controlling yet more transcription factors. It's also controlling cell cycle. It's also controlling signaling. So it's a very, very high upstream hierarchical factor controlling many downstream pathways. So that tells you that gene expression is disrupted in a global way, so that you have about a total of about 5,000 genes out of the total 30,000 genes whose expression pattern has gone wrong. Expression level is either too high or too low. But that doesn't tell you whether all these genes are directly controlled by the transcription factor OSMADS1. So for which we needed to understand what is its chromatin occupancy, what are the genes to which it itself directly binds. So we raised antibodies against a C-terminal domain of MADS1 that is specific to this transcription factor, and we dissected the panicles. Now remember, this is 1 millimeter. Okay, so sorry, the second M is missing, and this is 100 micrometer. So we have to dissect out these panicles which are embedded within the stem, and this is roughly about 45 days before any flowering is visible or seen to the eye. So we collect these tissues, and it takes us about 6 months to collect enough tissue to do one chromatin IP. So tissue is really limiting because we are targeting only the developmental stages. We are not looking at the differentiation stages. Then we make chromatin from them, pull down, do the Illumina sequencing, map the binding sites, etc. And we find very, very interestingly that MADS1, being a transcription factor, binds near the transcription start site, just upstream of the TSS of many genes. And that's about 12%. But about 40 to 50% of its binding sites are within the gene of its target gene, sometimes within the intronic regions, sometimes in exonic regions, and sometimes even in the 3' UTR. So these type of signature binding sites, that is within the gene and in the 3' UTR and in the far upstream, 3 to 4 kb away, are telling us that it's not just a transcription factor binding at the TSS, it's possibly binding at enhancer-like sequences, which are transcriptional cis-regulatory elements that can be very far away from the gene of interest, and by DNA looping can control the expression of the downstream gene. So MADS1 binds to about few thousand genes directly, but it regulates the expression of yet many more number, 5,000, 5 times as many. So then, using the DNA bound sequences, we asked what are the signature DNA sequences which we can understand from the binding site. So we are able to then query and ask what are the cis sequences present at the binding site. And this is a sequence element that appears in about 64% of the binding sites. And this is not a surprise. CC80 repeated 3 to 6 times, GG, is a signature that is bound by this ancient MADS domain. The MADS domain, 56 amino acids, that is found in yeast, it's found in plants, it's found in mammals. This is the signature sequence that it should bind to. So this sequence was found in about 64% of the MADS1 binding site, but there was a remaining good 36% of the genes. The binding site did not contain this cis element. Instead, it had only the central portion, which is called the AT tract. So majority AT tract without the flanking CG is found in a large percentage. It's not an ignore, you cannot ignore 36% of the binding sites. So then we were able to, with our collaborators, able to figure out what does the presence of an A tract mean? Does this define a DNA geometry? Does this define a binding site by means of DNA geometry rather than DNA sequence? And Manju and her collaborators were able to show that the region, this is the binding peak, the region around the peak is always less stable. The average free energy of the binding site region and 50 base pairs up and down of it is always less stable than the sequences which are outside. And so therefore, DNA geometry and free energy is also a feature that is recognized by OSMADS1. So this allows you, by looking at genome scale data, to discover new motifs which might be driving the binding pattern of a transcription factor. Now that we had the chromatin IP, which tells you all the targets that it is bound to on the chromatin, we needed a mutant which was a true genetic mutant. So we collaborated. Making a true genetic mutant in rice is now much easier with CRISPR-Cas9, but it was the traditional way of making genetic mutants by homologous recombination in plants is very difficult. Homologous recombination is very, very poor efficiency in plants, and therefore it was a big, big challenge. But Professor Velutambi at Madurai Kamaraj University developed a double strategy of negative positive selection. And even though it was very low frequency, we managed to get a homologous recombination based true genetic null, which was a deletion disruption. So this null mutant, again, all floral organs are abnormal, just like our RNAi. Too many floral organs because this meristem is continuously alive. It doesn't die after making a carpel. It keeps on dividing. The stem cell pool is alive. So we had this genetic mutant. So we combined the genetic mutant with the ChIP-seq to understand how does OSMADS1 control floral development, right from the point of making a floral meristem to the organ primordia. So shown over here are gene expression clusters in the wild type as development is going on. So this entire panel is only wild type. So as early florets are being made, there are a set of about 4,000 genes whose expression is very high and their expression goes down. This is another set of 3,000 genes whose expression pattern is opposite. So these are co-expression clusters which define how development progresses from early florets to middle florets to late florets. So this set of genes expression goes down, this set of genes expression goes up, this set of genes expression is high in 2 stages and then dramatically goes down and so on. Once we have these co-expression clusters, then we asked what is going wrong in the mutant. Does OSMADS1 control cluster 1, cluster 2, cluster 3, 4, 5, or 6? Or does it control all of them? And the answer is that yes, it controls genes in all the different clusters. Here is an example of cluster 1. What's happening in early stage of floral development is the meristem is growing while it is making enough cells to make the organ primordia. It is activating about 1,200 genes. And in cluster 2, it's activating about 400 genes. And in cluster 3, about 160 genes. But at the same time, it's also repressing these cluster 4, cluster 5, and cluster 6. So in early floral development, it's activating about 1,500 genes, but it's also repressing 1,500 genes. So the same transcription factor is controlling its target gene expression status both positively and negatively. That means its partners and the chromatin status is critical to its executing its function. So these are questions and challenges for the future. Now in this early set, what were key standouts were the set of genes which encode the homeodomain transcription factors. Homeodomain transcription factors, particularly OSH1 and OSH15, the reason they are very, very interesting, these are the homologs of the mammalian Yamanaka factors. So these are the pluripotency factors, and MADS1 is activating the 2 pluripotency factors, OSH1 and OSH15, in a feed-forward network. So in the flower, it is keeping the stem cells alive. It's keeping the pluripotent growth center alive so that more and more cell division is occurring to give rise to enough cells such a way that using those cells, organ primordia can then come up. So a feed-forward network is deduced from our experimental data sets. And even more interestingly, this feed-forward network controls 53 other transcription factors and chromatin modifiers. All these outer nodes are transcription factors and chromatin modifiers. And even more importantly, the blue are activated and the red are repressed. And only the filled circles are the ones which are controlled by the Arabidopsis ortholab. So the Arabidopsis closest relative of MADS1 is SEP3, and between SEP3 and MADS1, there are very small number of downstream targets that are the same. So as the two transcription factors have evolved away from each other through the time of evolution, their downstream targets and the mechanism of their activation has which is called diverged. Right? So this is a network which we have deduced using our experimental data sets. And we're also able to say that MADS1 in the early stage of floral development, it maintains the meristem and grows the meristem by this feed-forward loop. But the same MADS1 then switches to repress the growth once the floral organ primordia have begun, so that no more new floral organs can be made and the meristem is then terminated. Right? So a question asking, how does a transcription factor which is related to the Arabidopsis transcription factor have a unique phenotype just by itself, whereas in Arabidopsis, it's a group of redundant factors, led us to discover more and more and more questions. And now we know that MADS1 activates as many genes as it represses, and the sets of genes that it activates is different in time and space. So how does OS-MADS1 activate genes? It must be working with histone modifiers, which allow for chromatin opening. How does it repress genes? It must be working with other sets of modifiers or transcription factors which allow it to compact the gene. And all of this is happening dynamically in time and space. So it's a very, very complicated network in which we have got snapshots. What I'm showing you are 1 or 2 snapshots which allow you to make precisely this flower of the rice. Right? In the last, if I have 5, can I have 5 minutes? I'll give you another bit of a story on how transcription factors are evolving. Ancient proteins evolving all the time. So the second organ in the rice flower is called the lodicule, which I told you is the equivalent of the petal. And what is special about it? What the specialty is, it's a very, very short organ. And I told you that it's not present in a concentric ring. It's present in only 1/2 of the flower. In Arabidopsis, it's present as a concentric ring. The Arabidopsis petal is long but very thin. It has very, very few cells between the 2 epidermal layers, literally only 2 cell layers. Look at the rice lodicule. It's very thick, but it's very short. So it's a thick, fleshy organ with many, many cell layers and a lot of vascular bundles running through them. And the role of, this movie is not working. But because it is present on one side of the flower, the lodicule is present over here. It allows for the flower to open, the stamens to emerge, pollination to occur, and then it closes back in again. So I'm sorry, the movie is not showing, so I can't show you the flower opening and closing. But rice flowers open only for few hours in the day, and then they close back again. The opening allows for the stamen elongation, pollination, fertilization, and the closing is required for seed maturation and embryo growth. So if you don't open, you have a problem with pollination and fertilization. If you don't close, you have a problem with the seed drying out. So opening and closing in the correct time of the day is controlled by the lodicule. So it's kind of shown over here in a schematic. This is the swollen form which pushes the lemma and the palea out, allowing the stamen to emerge. And you can see the stamen has come out here. Here's the stamen after it's fully come out, but now it's closed back in again after anthesis. And this allows the fertilized ovule to give you the egg, or to give you the seed. Right? So the formation of the petals is controlled by B function genes together with A function genes. And in the case of rice, the B function genes are a single MADS16, which is equivalent of AP3, but 2 pistillata orthologs, which is MADS2 and MADS4. So in Arabidopsis, there's only 1 AP3 and only 1 pistillata, whereas in rice, there are 2 pistillata-like genes. So why would you have 2 pistillata-like genes in Arabidopsis? Is only 1. So these were answered by making specific mutations in one of the paralog or the other and asking what is the consequence of function. So if you lose the AP3 gene, then you lose the ability to make the lodicules and the stamens. Instead, you get a proliferation of extra carpels and bloom-like structures. If you lose both the pistillata-like genes, it's as good as losing the AP3-like gene. And again, you get only carpels and you get extra blooms or lodicules. That means you lost B function completely. But if you lose only one of the pistillata-like genes, then the lodicule is normal, but the stamens are a bit malformed. If you lose the other pistillata-like genes, then the lodicule is very elongated, but the stamens are kind of normal. So losing one, you lose one organ, you losing the other, you lose the other organ. And when you lose both of them, we get a new phenotype. So these were discoveries that we made that two paralogous rice genes can control new functions. Right? So we discovered that MADS2 has a predominant role in lodicule development. MADS4, it supports MADS4 for stamen development, but both MADS2 and MADS4, when we knock out both of them, in Arabidopsis, you have pistillata loss of function mutants are female fertile. But in rice, we saw that they were female sterile. We saw the ovule did not have any, this is a wild type, nice ovule with an embryo developing in there, with the egg apparatus, which will go on to make a proper embryo. Here, the egg apparatus itself is not there. There's no female unit, or there's just one egg cell, one polar cell, one egg cell, one synergid, and completely infertile female unit. Arabidopsis pistillata and AP3 mutants are female fertile. So that means B function genes in Arabidopsis work only for petal and stamen development. But in rice, B function gene is also contributing for female development in the carpel. So there's an expanded role for the B function genes in the fourth whorl in the case of the rice flour. So what is it doing? Is it even expressed there? So by doing RNA in situ hybridization, we were able to show that even though both the genes are contributing to carpel development, their expression profile is very distinct. MADS2 is expressed in the ovary just outside of the innermost wall of the ovary, right around where the integuments of the ovule are. But MADS4 is expressed inside the ovule itself. So even within the carpel, two closely related paralogous B function genes have spatially very distinct profiles. So these factors are evolving to have their own distinct temporal and spatial expression domain, and therefore together they are able to control female fertility, and each one of them are possibly controlling distinct set of genes within these carpal tissues. So we went on to do chromatin IP to understand what sort of downstream targets that it's controlling. But I will come to this one slide to summarize what MADS2's key unique features are. Remember, what is unique about the lodicule is its shape. It's a small organ which has a lot of cell layers in one axis and less growth on this axis. How does it have that so many cell layers in one axis? So it must be controlling cell proliferation in a spatio-temporal way. And what we found, all these blue arrows are cell division cycle genes. So MADS2 is directly regulating PCF2 and PCF5. This is proliferating cell nuclear antigen. PCF2 is the one which gets loaded on to the MCM at origins and allows for DNA or replication origins to fire. So two of the PCFs are directly controlled by MADS2. Yet others are cyclin D5-2. Then this is another PCF. This is cyclin D5-1, cyclin P4-1, cyclin D6-1, and there's also a CDK which I've forgotten to mark. So MADS2, which is controlling the shape and size of the organ by controlling where cell proliferation should occur, is directly controlling cell division cycle factors. The other unique thing about the lodicule is that it has to swell. When the lodicule swells, it pushes the lemma and the palea open. How does this organ swelling occur? This occurs through osmosis and quick intake of water through the vasculature that it has. So many of the genes which are required for vascular development and water transport, these are aquaporins and ion chimers. Either they are water transporters or ion transporters or peptide transporters are marked here in the red arrow. By directly controlling transporters which control the osmotic potential of the organ, it can cause the organ to swell or collapse. And this swelling or collapse makes the lodicule to function like a mechanical lever, allowing the flower to open and close. By directly controlling cell division cycle genes, it is controlling where cell divisions go to occur in order for you to make a thick parenchymatous layer with embedded vasculature present in there. So these were discoveries in just trying to understand how pistillata genes control the petal-like organ that's called the lodicule. And the earlier slides were summarizing that the pistillata type of genes are not only doing petal and stamen development, they're also controlling genes in carpel development. And our ongoing work is trying to understand how pistillata genes contribute to female fertility. So our journey into what we thought were class B genes has expanded our understanding to tell us that class B genes are contributing to lodicule and stamen development, but they also are involved in carpel development. We've also discovered that one of these pistillata genes is controlling flowering time. That is, even before flowers are made, when should you move from vegetative to reproductive development? This is again not a function that was predicted because we got the loss of function mutant and we're seeing loss of function causes early flowering. We know that this transcription factor is doing other jobs earlier in development. We think that it is involved in coordinating parthenocarpy, that is embryo development together with the ovary wall development to make a seed which is good, fertile with a nice embryo. You need to coordinate embryonic development with the endosperm and seed development that has the nutrition in it. And that coordination is done by the pistillata genes. How it is controlling these processes, flowering time, female fertility, preventing parthenocarpy are all big questions that we hope to answer in the time going forward. I've been absolutely blessed to have a fantastic team of students who've been working with me on this project involving rice developmental biology, of which in the beginning I knew absolutely nothing myself. I just had the courage to be able to say that if I am going to be working in India and I should be working on something which is unique and distinct and still within the domain of developmental biology, what would I do? This was way before it was rice was genome was known. It was not at all fashionable to be working on rice. It was absolutely rocking fashion to be working in Arabidopsis. But to walk away from the high life and to say that no, this is a new domain, and here is where I think I can make discoveries which will be pertinent. And there are questions which remain to be asked. It was that, I don't know, foolhardiness or daring, and having a team of students who believed in that, yes, if the tools are available, we can keep on deploying them to ask this big question that allowed us to make the progresses that we have made. Many of these are, very renowned professors in their own right at various institutions in India and abroad. You will recognize some of the names over here. And with me, these are the students who are currently pursuing their PhD. Some of them are very close to completion. One or two of them are having their exam in the next week or couple of weeks or months. I've had fantastic collaborators in India, abroad, and funding support from IISc, of course, the Department of Biotechnology, the Department of Science and Technology, and I can never forget to thank Rockefeller Foundation. So having an idea that, yes, you know what, we don't know anything about developmental biology of flowering and rice. Let me get into that is one thing. But somebody who will fund your idea and give you a research grant when you have zero publications in that domain, but know that you can deliver and fund you for it, that's Rockefeller Foundation. So they said, if you are working in rice, we will fund you, but we will not fund your Arabidopsis research. So they believed that I would do it, and I'm forever thankful for that. Thank you so much.
So thank you, Professor Vijayraghavan, for a very nice talk. Questions?
How MADs are regulated? So we know that MADs are regulating thousands of genes, but
Yeah.
what regulates MADs?
Other maths and many other factors. So I told you, for example, MADS1 is expressed only in the floral meristem. It's not expressed earlier on. So there, its own cis regulatory elements. So in the case of MADS1, it has a 5 kb intron. So there are elements within the intron which are contributing. So they themselves have to be controlled in time and space. So in Arabidopsis, the SEP3 genes, which are the equivalent of MADS1, are activated by LEAFY, which is a floral meristem identity gene. So each one of them is activated in a series of cascades. We do not know all the members which are activated, how they are activated. But they have to be activated in the right time and space.
So I have a question. When you are saying that MADbox protein binds to different region of the chromatin, right? So when it is activating and when it is repressing the same gene, does it bind different region?
Generally, it doesn't activate and repress the same gene. It activates one set of gene and it represses other set of gene, right? But the binding site will always have the carg element or the AT track. It is the partners which will determine what the outcome on gene expression will be.
Chromatin remodelers.
Yes, or other transcription factors. Its partners. So we know co-occurring with that, the frequency of occurrence of TCP domain factors, frequency of occurring of BZIP factors, binding sites is high. So we know there are at least 6 to 7 other cis motifs which co-occur with MADS1 binding motif. So which combination of that plus the chromatin modifiers will determine the gene outcome.
Usha, have you looked at, have you thought of doing some sort of IC experiments to see, in absence of this, what happens? Because
Yeah.
it binds to regions other than the promoters.
Right. So that we know, we haven't done 5C or even 3C or high C. We can do it, but we already know that it is making contact up to 4 kb away, up to 6 kb away, and so on. We can do it. We haven't done it yet.
But if you compare your ChIP-seq data with the expression like
that
the
we have
genes,
already done.
yeah, but do you see any correlation? Like,
Yes, that is how we deduced which are the genes that it is activating and which are the genes it is. Remember the co-expression cluster graph that I showed you? So in the wild type, 3,000 genes are going down with development. Of those 3,000 genes, 1,000 of them it is controlling. But whether those 1,000 genes are present together in the chromatin configuration, which is that we don't know.
so I was wondering whether you could do some sort of histone modification patterns.
Yeah, we can do.
And
That we
then
can do.
you see whether it's
Right.
repression and
Right.
expression is
So
same.
what I was thinking that we might do is we look at methylation pattern, look at methyl C pattern, or we will do nucleosomal positioning pattern using ATAC-seq. right? But like I told you, for any one seek, we need 6 months to collect the tissue. So, so first, before doing all of that, we have done it in the wild type context. So we can do chromatin configuration changes which are occurring and impose it on top of this. Yes, we can do it. So that would be attack seek or one of those methods. Yeah.
The other question I have is whether you can, uh, like, IPSC, can you make, can you reverse uh, a differentiated tissue,
Yeah, yeah.
multipotent tissue
This is what is happening
in
in our
plants?
mutant. Because remember, I told you that in the MADS1 mutant, you have an excess proliferation of organs. And within the flower, there is another flower coming. But all the organ systems are gone. So what happens over there is that the stemness
Yeah.
is maintained forever. In a flower, you should not make excess number of organs either. So the flower should make stamen, carpel, and stop. Within the carpel, it should make only ovule and stop. No more stem cells should be present. But in our mutant, we see that the stem cells are continuing. And because of that, it keep on making organ primordia. So you do see that. You do see that. Yeah. So if you do not terminate the stem cell pool, you will continue to remain pluripotent and keep making excessive organs, which is not good. Because the final organ system is the carpel with the ovule. After that, nothing more.
But once it is differentiated, you cannot reverse it.
We can.
Okay.
That's how people make callus. So plants are completely pluripotent. You can take, in the case of which tissue responds in different plants, it is different. If you take tobacco, even the leaf will respond. You cut the leaf, you generate a wounding signal, and you give auxin, then that causes cell proliferation without differentiation. So you get a mass of cells which are proliferated without differentiated. That's called a callus. And from that, you can regenerate somatic embryos, which are called somatic embryogenesis, through self-organization of growing centers. So that can be done in the case of tobacco, right from the leaf also. In the case of rice, it is the scutellum or the young leaf which is present in the embryo that is very embryogenic. Different tissues have different ability to show this totipotency or pluripotency, but it can be done. It can be done. They're very plastic. Yeah. So that is another model to study. How do you make a zygotic embryo from male-female gamete fusion? And how do you make a somatic embryo, which has come just from proliferating cells, which organize a center of growth and give rise to a new plant? So Kalika Prasad works on that. He's at Pune. Yeah, I said Pune. Yeah.
Ma'am, you mentioned that matchbox transcription factors might play a role in the opening of flowers in rice by regulating the vasculature. So I was wondering, how is that? I mean, we know that rice flowers open at a very specific time point, like
Right.
around 10 to 12 o'clock.
Right.
So
No,
how-
actually, even more earlier than that.
yeah, that's species specific.
Right.
Yeah. So my question is that how is circadian rhythm is coordinated with MADbox or maybe circadian rhythm proteins like high five or something like that is actually controlling the MADbox transcription factors in order to opening that flower?
No idea. Excellent question. So we haven't even begun to undo. Now we know what are the targets. Okay. But the targets, those targets must be also under some sort of circadian or other control. So our targets are developmental targets. They allow you to make the structure, right? If you don't have the structure, you can have all the circadian rhythm, nothing will happen. So you need to develop and get the right anatomy and the physiology and the right structure. But that structure has to function under circadian control. So that's another question.
Another question.
So all the targets of MADS2, one can now see how do they change in circadian. Right?
So, and ma'am, another question. in the developmental pathway, we see that certain RNA, microRNAs regulate the developmental process. Like 178,
Right.
72 goes up and 156 goes down.
Right.
So is it at the upstream of the Madbox domains, or is it at the downstream of the Madbox domain?
So, see, MADS domain transcription factors in rice itself are 50 plus. I talked about only the ones which are working in floral development. The 156 and 172 are upstream of SOC1 and all that, which is early in development. Got it? MADS domain transcription factors are, it's the Hox family of plants. So they are in almost every developmental process. The one which I talked to you about is downstream of 156, 172. 156, 172, then comes SOC1, then comes these genes.
Thank you.
Hello. I have a query that you have talked about the MATS is regulating a set of transcription factor for a particular upregulating or for another site you are basically replacing. My query is that whether there is an extended connectivity of the transcription factors which actually plays the role in regulating the others, or is the only the MATS one?
No, way be extended. It is not MADS domains controlling only MADS. MADS domain factors are controlling TCP domain factors, it's controlling BZIP factors, it's controlling MIB domain factors. It's not controlling only within its family. Its targets are transcription factors belonging to many different families.
Yeah. So my query is that whether there is any set of transcription factors, not only the one, which are actually controlling the others. It's a tight like that.
Yeah,
the feed-forward loop we have talked about.
Yes,
Whether
so-
there is a connectivity of the different kind of feed-forward
So
and
that,
feedback
that
loops.
this feed-forward network is between one MADS factor and two homeodomain factors. That is one feed-forward network. There are many such networks. We can show you only snapshots of it because that feed-forward network itself becomes a negative network later on. So that is between one MADS factor and two homeodomain factors. Between the MADS factor and TCP factors, there'll be another network. Between MADS factors and this, there are many other networks. Yes.
Is there any plan to look at the rewiring which happens within the transcription factors?
Yeah. So that rewiring is what we can only give you snapshots of it. But that is a dynamic process. Right? So if I can, in temporally itself, it is rewired in the normal. What is happening early should progress to the late, should progress to the very late. You understand that? That is in the wild type. In the mutant, what should be happening early itself is faulty. That is, right in the beginning, the wiring is wrong. So everything ensuing is wrong.
Sorry.
Why monocots will have to be so different than dicots in terms of complexity?
No, everybody is complex.
No.
So
It seems that monocots are much more difficult to work with than the Arabidopsis.
No, I beg to disagree. There are 350,000 families of extant flowering plants.
I used to
And
work
we-
with rice. Usha?
we work with, I know, we work with one or two species only. There are many difficult dicots and there are many difficult monocots. I think each species, it's evolved in its time and space.
In terms of these that you described, it looks to me, it looks that monocots are really more difficult to work with in terms of the regulation as opposed to the
I'm not sure because
dicots.
see, we are comparing even in dicots, we're comparing with Arabidopsis is only one member. I would argue that perennials are difficult, trees are difficult. You got it. Genome sizes make a P is great. Mendel discovered
The
the laws.
structurally monocots are very different. Like the ones,
Yeah,
our structure is entirely different
No,
than
no,
the
that
ones.
also leaf structure is
Yeah.
very different. But if they too, it's like asking why do we have fish versus mammals, right?
I
So
just,
they're
I just,
structurally very different.
I s-
That happened 120 million years ago.
I
Why
see.
that happened, I don't know. But it has happened for a good thing. Otherwise, we will have no energy, no calories.
Usha, I just wanted to get your comments on it.
I don't think that any one species is more difficult than the other. It's our ability to develop the methods to understand it. Both are difficult in their own way. Arabidopsis is easy because of the short lifespan and small genome. So therefore, you can make faster progress over there. But that's not always the case with all dicots. They come with their own challenges. Each one come with their own challenge. It's impossible to do the kind of genetics we're doing now. You can't do it with P. But we understood Mendel's law for entire life using P. But we cannot study P very deeply because it has its own inherent problem. Genome is huge. Same way. So it is, I mean, different plants come with their own plus and minus advantages. Same thing with wheat. Yeah. Thank you.
So I presume.
Hello. So some homeodomain containing factors are also stress responsive.
Yes.
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 So
yes.
do they share some common cis elements, or but how they differentiate between on the which part of that?
Right. These two, which are the OSH1 and OSH15, which are the stemness factors, they are not involved in stress. In stress, yet other factors are. So again, it's a large number. There are about 30, 40 members of each of them. NAC domain, there are about 70 of them, 70 plus. So some members are involved in stress, some are involved in development. It's not easy to predict which one is which unless you start doing the functional studies. Right? The number doesn't mean anything. Number is just when it was discovered.
So if there are no further questions, let us thank the speaker. We'll have a very brief felicitation of the speaker by our director. So